Welcome to the practical demonstration on agarose gel electrophoresis. All laboratory procedures were conducted at the Molecular Biology Laboratory at Spectrum Institute of Science and Technology. To perform agarose gel electrophoresis, we will require the gel casting tray, combs, the voltage unit and the gel tank. Before we commence the agarose gel electrophoresis, we have to prepare the agarose gel. The agarose gel is prepared by dissolving agarose on the buffer solution, which can either be the tris acetic acid EDTA buffer or the tris boric acid EDTA buffer. The solution is prepared by heating and is poured on the gel casting tray where the combs will be inserted. Once the gel solidifies, the combs have to be removed very carefully. Make sure that you do not damage the wells while removing the comb. You remove the excess gel off and then you put your gel inside your gel tank. Make sure the gel is completely covered by your buffer solution and all the wells are completely dipped in the buffer solution. When loading, you will require to add your loading dye before you add your sample. So here you take the required volume of loading dye, which would contain the loading buffer, which will allow the DNA to settle inside the well by adding a density, and the tracking dye, which will allow the visualization of the migration of your DNA during the gel run. You also can add a DNA ladder, which is a predefined ladder containing fragments of different base pair sizes. Here we are using a 1 kb DNA ladder that will separate into fragments with specific base pair sizes of DNA. When you add your sample onto your loading dye, you mix the contents very well before you load into the gel. This is a very important step as it will allow the mixing of all contents with the loading dye in order to be loaded into the gel. Once all the contents are taken, you carefully load it into the gel. Make sure that you do not damage the wells during your loading step. Once the gel is completely loaded, you will close the gel tank and then set your voltage and your time for your gel run. Once the time is set, you can begin the gel run, and when the gel run begins, you will see the DNA bands separating as per the indication given by the tracking dye. Once the gel run is completed, you will transfer the gel onto the UV trans illuminator for observation. Here the DNA visualizing agent, which is ethidium bromide, is used. Ethidium bromide staining can be done as 2 steps, either as pre-staining or post-staining. In this video, ethidium bromide was added during the preparation of the gel, which is the pre-staining technique. During post-staining, ethidium bromide will be added after the gel run. Ethidium bromide is an intercalating agent that intercalates between DNA and illuminates under UV. So here you see your DNA bands being illuminated under the UV trans illuminator. Thank you.